Next up is Michele Detry. Michele Detry is a biostatistician who works with Berry Consultants, works on adaptive clinical trial design, is my co-leader for section 2, and she's going to talk about multiplicities and the risk of looking at too many things.
Oh, yeah. That's super cool. Okay. I probably don't need it, but I'm going to use it just for fun. So there's this great title for my talk, Multiplicity, and it's like incentives of fooling oneself and the risk of something like that. I don't know why I didn't keep that title. It's much better than my title. Basically, we're just going to talk about multiplicities in clinical trials. So multiplicities, multiple tests is such a consideration in clinical trials that we've heard it in at least 3 other talks so far this week. Which ones? I like to be interactive on this afternoon. So what ones have we kind of talked about?
Multiple ones.
About what?
Multiple ones.
Multiple ones, good job. 3 talks already. Monitoring, exactly, your talk. We talked a little bit about sample size. And we talked about some in study design. So it's so important that was already talked about 3 times before I got up here. So a little bit of this is redundant. But I hope to give some insight and some personal experience. So the concern, multiplicities, multiple comparisons, the concerns arise when numerous statistical tests are performed. I'm not talking about summarizing data, looking at a bunch of your characteristics, summarizing means, standard deviations, frequencies, things like that. I'm talking when you do actual statistical comparisons and draw inference from the results. As we've talked about, some of these can be real. Some may be significant due to chance alone. You're not going to know which is which. So what we do is we try to reduce the probability of making one of these errors, and that's where we've talked about type 1 error a bunch. And so we set different thresholds that if we see a result that we saw, our result, or something more extreme, that the probability of that happening in the null situation, we want that to be small. The more tests you make, the greater a chance you have of making this error. So, so you can see how in a clinical trial where you're collecting lots of information, this could be a big problem. So, did anyone find that I put a background paper in with the background readings? Did anyone read it? Oh, it talks, it's kind of fun because it talks about you're traveling in a foreign land and you walk through Oz, and that's how the paper starts. So if that doesn't catch you and keep you right there, I don't know what will. So it's a paper that Don Berry wrote. It's in JNCI, and although it's written for a cancer journal, it's not specific for cancer. I think it applies across all disease areas. So I didn't want you to see that it was in a cancer journal and not read it. I think it applies across all areas. And what it kind of talks about is it's a good background. I think it's written well. It doesn't have statistical formulas all throughout. And it talks about there's a complicated nature in handling multiplicities, and there's different opinions. It's not clear cut of what you need to do when it's a problem. And this will give you both sides. And I think that's important because you should always think about multiplicities in the context of your research. And I also like the strategy it gives because one reason I like it is because I agree with it. And it's something I use. But I think it gives a nice bullet point at the end that you could always refer to as a reference to follow. And so hopefully you'll get a chance to read it. So how much of a concern are multiplicities, multiple tests? Depends on your research question. As you heard over and over again, so many things depend on what your research question is. There isn't this set box that you always fit in. Everything should always be tailored towards your research question, answering your research question, and taking it into consideration that way. It was already discussed by Dr. Yates that this 0.05 is not magical and doesn't fit everyone. 80% power doesn't fit everyone. Everything you think about is in the context of your research question and your research that's being done. So for example, we have a phase 3 confirmatory trial. This is something you're going to do. You've got a drug out there. This is the final step. This is the big confirmatory trial. You want to get this drug or this intervention used in clinical practice. You want to change clinical practice. You want a high level of evidence. You want where anyone can look at these results. They're going to be confident in the results, and they're going to say, okay, look, I believe this. I'm changing. I'm doing this treatment. You want a high level of evidence. Therefore, you're going to want a more stringent control of type 1 errors, and something being significant due to chance alone is going to be a big concern. On the flip side, let's say you have an exploratory study, an earlier phase study. Maybe it's something you use to kind of do some exploration and generate hypotheses. In this case, you may be willing to give up some control of the type 1 error because it's exploratory, but based upon what you learned in your study, you're going to have subsequent research to confirm those results. So you may be willing in that situation to have a type 1 error rate. It all depends. It depends on the context of your study question. One of the big things you need to think about is what's the next step. When you're done with your study, what happens next? In a phase 3 confirmatory trial, what happens next is people are going to go use the drug. That's a high threshold. Maybe if in an earlier phase study, you're looking for a dose or doses that you're still considering for the treatment, maybe you're willing to have a little more risk of making a mistake because you're going to have this next step to confirm it. So multiplicities, it's a consequence of lots of data. And we kind of heard about this before, that it's very easy to collect many, many things when you do a trial. You finally get your study funded. You're really excited. It was a lot of effort. You went through submitting grants. You went through the review process. You got IRB approval. So you want to, you kind of come from the sense of, I finally get to do this. I want to make sure I collect everything I need and not miss anything. This can be a problem because now you're going to have all this data. You have all this data. What are you going to do with it? You want to look at it all. I mean, you collected this. You want to explore it. So blindly going out and just collecting everything out of the universe is probably going to lead to concerns with multiplicities and all the things you're going to want to analyze. It's very easy to do today. It's very, with electronic case report forms, the ability to process lots amount of data. It's very easy to get all this data. Therefore, it lend itself to a multiplicity concern. And then once you have it, like I say here, it's very tempting to explore anything and everything to find something significant because you know that's what the journals want to see, is you think that the journals want to see, you think that your colleagues want to see significant results, and that's what they'll be interested in, that's what will get you published, and it's just very, very tempting. But you want your results to be credible, and you want things to be reproducible. So if you send this, if I read a paper, I want to be able to read it, and I'm going to decide whether I or not believe it based upon the credibility. I'm going to think about how many tests were performed. And it's also very important that if someone conducts a similar study, they get a similar result. Reproducible research is very, very important. And so this is where the talk we had earlier by Dr. Johnston about thinking about your case report forms and what you're going to collect and limiting it to the most important information that you really need is important, because then also you may be able to limit this temptation to go down rabbit holes looking for something that may not be real. So I'm going to give you 3 general broad examples of multiplicity that you may see in clinical trials. You may see multiplicity concerns, considerations arising from multiple tests across multiple treatment arms. There may be multiple tests amongst subgroups. And you may have multiple statistical tests performed while data is accumulating. So those are just kind of some 3 broad categories that we're going to go through some examples. So let's talk about multiple treatment arms. Let's say you have a randomized trial of 4 active doses of a new drug and a control or placebo arm, whatever's most appropriate. And you have these 4 active doses, and you have this natural tendency to want to compare each active dose to the control and see if it works. So in this case, that's 4 tests. Let's say these doses are ordered. Let's say they're 1 milligram, 2 milligrams, 3 milligrams, and 4 milligrams of some drug. Just keep that in the background. So you run the trial, you do these 4 tests, you find p-values of 0.20, 0.18, 0.04, let's say 0.049, and 0.30. When you get done, you look at these results, do you believe that dose 3 is the only effective dose? Do you think others will believe it? What do you think? How many people think yes? How many people think they may have concerns? You're testing at the 0.05 significance level.
I vote. I guarantee the vote is 3.
Guarantee the vote.
I would like to see confidence intervals.
Would like to see confidence intervals. Okay, so there's some. So the big thing is there's questions. There's questions here. You did multiple tests. You're really close. One of the other things that raises questions for me here is I had doses of 1, 2, 3, and 4 milligrams. I might have thought, hey, there might be a dose response here. Maybe as I increase the dose, I might want to see more of a response or more of an effect. And it's kind of weird that I had this one in the middle show up. So do you think it's real? Do you think it's random due chance? You don't know. So the question is, do you need to account for multiplicities here? think in the context of the question in the background. So again, this is where I thought about, I've got these 4 doses. I was kind of thinking going in that I was going to have a dose response. That's why I was looking at increasing the dose and hoping that maybe I have more of an effect as the dose went higher. So it was kind of weird here. So in this case, just simply going to compare each of the doses to the control really wasn't thinking in the context of my question in the background that I had. And that was just solely going for a focus of getting p-values. And as we've heard before, you don't want to blindly live just by p-values. So what I would have done is I would have thought about these issues and my study question to be very clear about when I was designing the study and writing the protocol. And one thing I would have done is I would have modeled the dose response. I would have thought there's some sort of dose response going on here. That was my hypothesis going in. And I would have used, so that would have helped use the information of all the doses together, thinking that there's some sort of curve across these doses. And I would have used that to develop a rule for how I pick what dose is best or what 2 doses are best. So I would have thought of this in a different context, and then I would have done some clinical trial simulation, which Kurt's going to talk about later, but then to see what my chances of making a type 1 error rate. I wouldn't have just naturally gone to compare all these to the control and then had this issue of multiple testing. So it brings up another question too. So let's say you have a company that has these 4 doses, 1, 2, 3, 4 milligrams, and you wanted to compare them all to control. If you split up your company into 4 smaller companies and ran 4 trials, all of a sudden all these trials can be tested at the 0.05 significance level. And so each trial you're doing 1 test, all of a sudden it's not as much as a concern. Why is that? Is that kind of confusing, thinking, well, wow, if I split this up into 4 different trials, now I'm not held to the same sort of standard. Go ahead.
Well, you've got 4 different placebo groups then.
You have 4 different placebo groups and you have 4 different populations. So you really, in that case, you had kind of 4 different trials going on. You were doing 4 different samples. So it really isn't a completely equivalent situation. But that's one of the arguments people usually say is, well, I can't test them all here, but if I had a different study and I just tested one of them, I wouldn't have been held to the same standard. So this gets into the complexity of the multiplicity concern question. It's not straightforward, but there are some guidelines that you can follow to make good decisions, to present your research in a credible way, and to have people read your results and have faith in your results. And by the way, please feel free to interrupt me at any point and ask questions. Questions? Okay, let's talk about multiple subgroups. And this one is really, really tempting to do. Let's say you have a randomized study and you're comparing intervention A to standard of care. At the end of the trial, you're really disappointed. You didn't find a significant result. I mean, you did this study, you thought it was going to work, you were sure it was going to work. And you went through and you're just really disappointed. This was a 3-year trial. You got done, you have lots of data. You're thinking this had to work, where did I go wrong? And so you start looking in subgroups. And you start diving in, you look at different categorizations of age, you look at gender, you look at different demographic factors, and you get down to a subgroup of males who are age greater than 50 and had some pre-existing disease X, and voila, we got a significant result. Do you believe it? It could be real. But the way you went about finding it, the fact that you didn't pre-specify it and have this as a hypothesis before you began looking at the data has weakened the credibility of this. So I would be hesitant to draw a firm conclusion from a result like that. Would you publish the result? You want to, I mean, it's tempting. You found something in this trial, you didn't have any other positive results. You want to publish it, but then the big question is, how do you frame the result in the context of the multiple subgroup combinations you examine? If you're going to publish it and you feel that there is a good rationale for this is something that might be real, and we talked about earlier this week that you can probably make an argument for anything. I mean, the more you look at it, you saw this result, you can probably rationalize, that's why it happened. But you need to be honest and state what you did. So if you're going to go and publish this, you need to say, this was a post hoc analysis, it wasn't pre-specified, I did 30 analyses of different subgroup combinations, and here's the process I went to before I got to this one, And this was the only one that showed the evidence of an effect with the interaction. Now, as a reader, that gives me the context I need to know how firm I should believe, how much I should believe this result, and also gives the research community, if it's something that they agree with, that there needs to be additional research to really confirm this.
I have a question.
Yes?
Is there a way to calculate the type 1 error then?
We'll get to that a little later, but you can do adjustments to the type 1 error rate to account for the multiple tests. But the more tests you do, the lower that threshold is going to have to be for statistical significance. And we'll talk about that a little later. So you're going to have a higher bar that you have to reach. So if I did 20 subgroups, there are many different methods, but it's going to be a much more stricter level of showing that there was a difference. And I'm going to have a big difference.
But for a particular post hoc analysis, like an example you were just giving, you say, well, I did these other 30 comparisons, considering that I did 30 post hoc comparisons, now the type 1 error rate on this could be such and such percent.
And you can compute that and put that in, and compare that to the evidence that you have from the statistical tests you use, the probability of there being a difference. And I don't think that's the strategy to go through, but I'll go through it later. So here's another example. Let's talk about multiple testing. So here's one example, and a lot of these are things I've seen throughout my collaborations with researchers. So this is more maybe an observational study. I'm working with a researcher. They've run their trial. They had half the subjects enrolled and half the data outcomes collected for those half the subjects. Didn't plan ahead of time that they were going to do any sort of interim analysis to look at the data. But an abstract deadline for a conference you really want to present at is coming up in 2 weeks. And you really want to submit something because when the conference happens, your trial will be done. So, do you analyze the data and submit an abstract if you see a positive result? And I apologize if you think these are silly examples, but they're all examples I've seen. And situations that if you haven't already, you may go into. Should you just go look at the data, analyze it, see what the result is, and submit your abstract? And then maybe when the conference comes, you will update the information in the abstract with the full results? Do you want to do that? Someone's got to answer. Otherwise, I'll start calling out our group too. No, why not?
Because it impairs your statistics for your final result.
You have the multiple tests, which is true, but the other thing that I would be most worried about, well, the unblinding, obviously, if the blinding thing, that's a big thing if you're unblinding yourself and you weren't supposed to. What if your results change? So you submitted this abstract and you didn't take into account that you were going to do this early look in the data, you didn't take into account this multiple testing, you got a result, you submit an abstract, got accepted, you're doing your posters, you analyze your data again, you've got a totally different conclusion. And now your abstract doesn't match anymore, and that's a very real risk. You designed the study to have a certain full sample size at the beginning for a reason, and you didn't plan to have this middle look. So you have a big risk going on there. So there's that could be a big problem. Another one, and this is another example I have, multiple testing. You have a trial with a continuous outcome. I don't know, some sort of blood pressure measure, cholesterol measure, I don't know, whatever you want to make this continuous outcome be. You have a blood level that you're measuring, and you're thinking that you may have a difference between these two within some categorization of these blood levels. So you're going to take this blood level, whatever it is, and you're going to categorize it good and bad. And you want to look and see if there's a difference between your outcome between these two groups. So you're not quite sure what the best way is to categorize this blood level. So we talk about it, and you look at the lab levels and the upper, you look at the upper limit of normal for something that's high versus low, and you use that, and that's how I'm going to cut point. Use the cut point. Do the comparison the first time you do the task, it's not significant. Researcher goes back and says, well, that might have not been the best characterization. Maybe I should use this other cut point. I've got some literature out there. It might be a reasonable, I can back it up what this cut point is. Do the test again, still not significant. But it was closer. You got that. At that point, I did have a situation where I was collaborating with a researcher, and they said, well, how about we try this other cut point? Let's try this third one. And do the third, let's say in that situation you would have done the third test and it was significant. Do you report the difference just using that third categorization of the variable? I'm sure it's been done. I'm sure it's happened in the literature. And this was a situation I had where I was concerned about the credibility of the results, And I explained to my collaborator the concerns with multiple testing and the risks that they were taking. And then I also explained to the researcher I would only do the third test if they reported the other two alongside of it. And they decided at that point they didn't want to anymore. So my job as a statistician is for the credibility of the results, to help you answer your question, to do everything I can knowing of these problems ahead of time. I can say they probably weren't very happy, but I think it was the best for the science in answering their question. My job is not to be a barrier, but to defend the credibility of the research. So that one's a big problem, and you may see, and it's something that's very, very easy to do. Third example, you've got, and we kind of talked about this, you've got this large double-blind randomized clinical trial. It takes 3 years to enroll. You've got 2 arms in intervention and control. Your outcome is measured at 3 months, so while the trial is ongoing, you have subjects that finish their follow-up. You want to look at the data unblinded during the trial to see if you can stop early for success. So we kind of had this example earlier in the week where we talked about you get these multiple interim analyses, you've got these multiple tests. So you need to think in this similar table, mine's kind of an abbreviated table, but there is this paper in 1969 that talks about the number of tests that you do, the number of looks that you do in a clinical trial, interim looks, the probability of making a type 1 error increases. So you need to control this somehow. You don't want to just blindly do these tests at the same significance level, not take into account that you're doing multiple looks throughout. So what you do is some of the techniques that were discussed, and so you have to think these early and interim analyses are going to be are maybe different than the later interim analyses, and you want to be really careful about stopping if you're not really sure what's going on, because you have smaller sample sizes and less evidence at that time. So the question is, how do you handle these interim analyses and the resulting decision? So there are acceptable, well-known methods, and there are new methods being created often to try to address different interim analysis plans, different goals of doing interim looks at a large clinical trial while they're ongoing. It requires a lot of pre-planning, but I say that's always good. It's always good to do the planning up front and make sure you have a well-designed study, you know what's going to happen, even if you don't have interim looks at the data. So that's not a bad thing, but it does something just know it requires. And it may require a higher level of evidence at the early interims than the later interims, because you want to be really sure that you've got a benefit if you're going to stop early for success early in your trial when you were planning to go to the end. And so that was kind of the description. Dr. Challoner, if I mispronounced your name, I'm sorry, talked about the different spending boundaries, the different boundaries you had to cross in the clinical trial to stop early, and there was a higher threshold in some boundaries earlier on than later. Some of them were flat throughout, but there's many different options, and you take that in the context of your question and what you're willing to consider for stopping early. So when you do this type of analysis or this type of design, you must pre-specify complete details of the design. And so this is kind of just, since I brought up adaptive designs, looking at unblinded interim analysis, I feel like I always have to say this even though this isn't exactly multiplicities. You need to think about when are these interim analyses. You need to specify ahead of time what decisions are going to be made during these analyses, so it's not subjective when it happens. You need to account for the type 1 error increase through the multiple tests, and that can be done through many increase. You need to think about who performs these unblinded analyses, because you don't want to unblind the clinicians of the study team. You've got to have someone separate. And there are many other logistical considerations, but I just have to throw it in. And trial simulation is very important, and Kurt's going to talk about that later, so I don't need to. All right, so let's go through, and I've got 4 minutes till break. Let's go through another example. Let's say you have a clinical trial for traumatic brain injury. You're examining some experimental treatment versus standard of care. You're not sure how soon the treatment needs to be administered, but you think sooner is better. So you think if you administer this treatment within 2 hours after injury, and this is a completely hypothetical example, that's going to be better than 2 to 12 hours after injury. But you're not sure, so you don't just want to look at 0 to 2 hours. You also want to enroll people that maybe are a little longer out, because maybe you'll see a benefit there too. So what you want to do is you want to compare the active treatment to the standard of care in both populations. So let's say this is a later stage confirmatory trial and you have 2 tests now. And so we need to now take into account that we're doing 2 tests in these 2 populations, the within 2 hours and the 2 to 12 hours after. So I'm going to start, and let's say I was targeting a type 1 error rate of 5%, but we have these 2 tests, so we have this increased chance of making a type 1 error. Do we have to adjust for the multiple tests? In this case, because of where you are in the research and you're really wanting to change practice, I would say you need to consider an adjustment for the multiple tests. How many people have heard of a Bonferroni adjustment? Okay. So, and if you haven't, what that is you take the significance level, the 0.05, the alpha, and you divide that by the number of tests you're performing, and that's the new significance level you're using for all the tests. So in this case, we had 2 tests, and so if we did a Bonferroni adjustment, we're now down to a 2.5% type 1 error, 2.5% alpha. The p-value has to be less than 0.025. But as we talked about before, when we were talking about sample size considerations, now because we lowered the type 1 error rate, we lost some power. If we want to get the power back up to where we really had to have the adequately powered study, we have to increase the sample size. So there's a consequence here. So you need to think about, we've got these 2 tests, you know, I really, really want to do them. Okay, that's great, but there is a consequence to that. And it might require a larger study of more subjects in each group. But we haven't thought at all yet here about the question. You think that giving the intervention sooner might be better than giving it later. So let's say you do the test in the 0 to 2 hours group and it's not significant. Do you think there's really going to be an effect in the 2 to 12 hours group? Probably not. It goes against all the background you have, all the models, all the preliminary data. I mean, you were thinking if 0 to 2 worked, maybe 2 to 12 would work. So maybe instead of doing this Bonferroni correction, which is kind of harsh, maybe there's something else I can do. And so the example I'm showing here is just a example. There's lots and lots of different examples. There's a whole literature on adjusting type 1 error rate. But in this case, I might want to consider something called a gatekeeping option, where I start and I test the active treatment versus control in 0 to 2 hours group. If it's significant, then I go test the other group. If it's not significant, I'm done. So what that allowed me to do is do the test at the 0.05, keep that same sample size that I originally wanted. I controlled type 1 error rate. And it was, the point I wanted to make here is I thought about what I was going to do in the context of the question. Everything's in the context of the question. This had to be pre-specified in the protocol. You needed to follow it, follow your plan when you got done. But that is a potential strategy that would be accepted. So how to handle multiplicities. My #1 recommendation on how to handle them is to put much thought in how you define your study question. I imagine if your small groups were similar to mine, we've had lots of discussion about study question, really teasing that down to be focused. If you really work hard on defining your study question, making it focused, selecting a primary aim and a primary outcome, you can reduce this to maybe a single primary, 1 outcome, 1 test, and be focused and have additional analyses be secondary exploratory. But have that one question, which is always, when I get done to the trial, how am I going to say if I won? What's the decision I'm going to say if I won when I was done? Now, there may be situations where multiple tests are completely warranted and you need to think about adjusting for it, but if you can get a single goal and be very focused, that's going to be easier. Not easier. That's going to be, you're going to be able to use your, not have as strict a threshold of adjust for the type 1 error rate and all the consequences that might go with that. You can have other aims that can be secondary and exploratory, but they may not have the same level of evidence required. And they're going to be reviewed that way by people reading your paper, reading your report, looking at results. Pre-specification is key. You need to think about what you want to do ahead of time. Anything you start looking at afterwards is going to have less credibility because you have the data in hand. Once you have the data and start looking at it and start doing things you didn't plan, I'm more suspect in the results that they might have been due to phishing and just kind of going through as many possible combinations as possible, and maybe if you didn't consider for multiple tests. So define your primary and secondary analysis in the protocol. Describe how you will handle multiple tests, and if you're not going to have adjust for multiple tests, you need to defend it. You'll see in the paper I sent, there's actually an argument that Rothman made for doesn't believe that you should have to adjust for multiple tests. I would say that's not, it's a compelling argument. I can't say I disagree with it, but I can say in certain situations, like the level of evidence required in a confirmatory trial, that I wouldn't want to rest on doing as many tests as I wanted. I would want to have that extra level of evidence, that extra level of security, that I would have credible results when I'm done. When you're reporting results, you need to be clear on how you arrived at the results. Was it the primary analysis, or was it an exploratory multiple subgroup analysis? How many subgroups did you examine? Were these subgroups predefined? Interpret them correctly in the context of what you did, be honest, and do not overstate. So the worst thing would be to publish something, not be forthcoming with what you did, and then have other people try to reproduce your results and get a completely different result. And then it comes forth that, oh, you maybe did a little searching from this and this wasn't pre-planned. There are different statistical methods for adjustment for multiple tests. It usually requires more evidence and a higher probability that the result is real, a lower possibility that the result is due to chance alone. But the main story is limiting the probability of the result. The main story with these adjustments is you're limiting the result that the probability can be due to chance alone. And one way you can do that is by having a very focused research question. Because if you do these adjustments, there are consequences. We just showed that little example where if I lowered my type 1 error rate, I lost power. To get my power back up, I had to increase sample size. And maybe that wasn't something I'm able to do. It wasn't feasible with the subjects that were available. It wasn't feasible with my budget. And so you need to balance your desire for multiple tests with the consequences or adjustments that you may need to make. So in summary, multiplicities is complicated. I can't sit here and say this is exactly what you do in each situation. But you need to think about it ahead of time. You need to be well aware of it could be a criticism when you're done. So you need to prepare your protocol in advance. You need to specify your primary aims. And you need to think about this if you're going to have multiple tests and some sort of primary outcome, or even if you're going to have 30 secondary outcomes that you're going to test. You're going to need to think about what doing all these multiple tests is going to apply when people read your report. In publications or summaries, be clear what was found through pre-specified analyses and what wasn't, be honest. You need to keep track of everything you did. It's very easy to go start looking at things and not keep track of where you were and how you got to this point. You need to have a plan ahead of time to keep track of what you did. And adjust where warranted. Better to focus during the design phase if you can, keep yourself focused. But there are methods to adjust. There just are consequences to it. And you want your results to be reproducible. If results from an exploratory analysis, you may need another study to confirm. So you always need to think of what's next. What am I going to do with these results? What next? What level of evidence do we need? And consider your results carefully in the context of the research question, the prospective biological rationale you had before your study, previous published studies. Maybe there's such a wealth of evidence out there that you can defend that this wasn't phishing, that this was something plausible, that maybe you didn't adjust, and you might be able to make an argument. It all depends. And if you prepare multiple publications, it's very common. You've got a clinical trial, you do one paper on the main result, you do another paper on this result, you do a third paper on this result. As I read paper number 3, I don't know how many other papers that were done analyzing the data from this trial. So if you prepare multiple publications, be clear about all the analyses and the other publications that were planned so people can get the context of what you did in the context of the full breadth of the analyses so they can consider the evidence and the possible multiple testing concerns. And so that summary kind of coincides with a summary and some other points that are in the paper that I put as a background, but that's what I have. And so if you have any questions, I'd be happy to answer them.